Welcome back, students. We'll start with yet another interesting topic in chemotherapy, antiviral drugs. So, the learning objectives for today's session will include to know about viral infection and pathogenic viruses, and second, to study the classification and actions of various antiviral drugs. We are aware that viruses are small infective agents which consists essentially of a nucleic acid, either an RNA or a DNA, enclosed in a protein coat or which is also called as capsid. Now the coat plus the nucleic acid core is termed as nucleocapsid, as you see it in the diagram. Now some of the viruses, in addition, they also have a lipoprotein envelope which may contain antigenic viral glycoproteins as well as host phospholipids, which are acquired when the virus nucleocapsid buds through the nuclear membrane or plasma membrane of the host cell. So here, although the structure of virus appears to be very simple, it's just having a nucleocapsid and an outer lipoprotein envelope. That's all. There is no other cell organelle present in the virus structure. Now, so what is happening here is now these intracellular parasites have no metabolic machinery of their own. So they have to, for their survival, they have to depend upon the host cell. So in order to replicate, they have to attach to and enter a living host cell. And this living host cell could be a animal cell or a plant cell or a bacterial cell, which is essentially required to use all its metabolic processes for its survival. Now, the binding sites on the virus. Now, let's talk about the binding sites. Now, the details of these binding sites, where the binding sites are, which virus acts on which host cell structures binding site, they're all given in detail in the next slide. So, I'm just going to summarize. These binding sites on the virus are basically polypeptides on the envelope or the capsid. Now, the virus specific receptors on the host cell to which the virus attaches are normal membrane constituents. It could be like receptors for cytokines or neurotransmitters or hormones or ion channels or some integral membrane glycoproteins. Now, all these examples of the host cell receptors and the details of which particular virus is it's acting on, it's all given in the next slide. Okay, so what we have known so far is viruses having a very simple structure. They are intracellular parasites having no metabolic machinery of their own, and they have to essentially depend upon the host cell for its replication as well. Now, talking about the types of viruses, there are basically three types of viruses. One is a DNA virus, second is the RNA virus, and third is the RNA retrovirus. The first, DNA virus. Now, DNA viruses are they are generally they generally the entry of the DNA viruses of the is of the viral DNA into the host cell nucleus. And once the DNA viruses enter the host cell nucleus, there is transcription of this viral DNA into mRNA by the host cell RNA polymerase. So what is happening with the DNA virus? Now DNA virus, for example, it could be examples of DNA virus, I'll tell you. They are pox viruses, which cause smallpox, or herpes viruses, which cause chickenpox, or adenoviruses, which cause sore throat or conjunctivitis, or papilloma viruses, which cause warts. Okay, so these viruses, what do they do? Once they enter the host cell and under the influence of the enzyme RNA polymerase, they are transcribed into mRNA. So viral DNA here is getting converted to mRNA by using the enzyme RNA polymerase. This transcription takes place. Okay, and this is followed by translation of mRNA into virus specific proteins. The second is RNA viruses. Now RNA viruses, what do they do? These enzymes, for example, now RNA viruses are orthomyxoviruses, which cause influenza, or paramomyxoviruses, which causes measles, mumps, or rubella virus, which causes German measles, or rhabdoviruses, which causes rabies. Okay, so these are all RNA virus, specific RNA viruses. What do they do? Once they enter the virus host cell, their mRNA will serve as its own mRNA. So this mRNA, there is no enzyme to transfer into mRNA because its RNA itself is serving as an mRNA, and this is translated into various enzymes, including RNA polymerase, which direct and which help in synthesis of more viral RNA and also the structural proteins of the virion. Whereas in case of RNA retroviruses, the virion contains a reverse transcriptase enzyme which makes a DNA copy of the viral RNA. And this DNA copy is integrated into the host genome and is now called as provirus. And the provirus DNA is now transcribed into the new genomic RNA and mRNA for in turn to be translated into viral proteins. So the examples of viruses which are acting through this way is the very common AIDS virus or the HIV virus. So we have seen that in RNA retroviruses, you have an enzyme called reverse transcriptase that plays a very important role in making a DNA copy of the viral RNA, and this gets integrated into the host DNA, now called as provirus, and this is ready for getting transcribed into mRNA and then into translation to viral protein structures and its enzyme for viral. So in this figure, you can see that how the virus enters the host cell and how it is acted upon by several enzymes for production or for multiplication into new virions. So the top portion shows a structure of a virus which has a nucleocapsid, enzyme protease, enzyme integrase, enzyme reverse transcriptase. So this virus is nothing but an HIV or a AIDS virus. It is also surrounded by an envelope. This appears that this envelope targets the CD4 glycoprotein or the helper T lymphocytes, through which it has access to the cell. Then there is a fusion of the virus particle with the cell membrane through which it is acting. It's finding its entry into the host cell. And the next comes the enzyme reverse transcriptase, this being an RNA retrovirus. The reverse transcriptase makes a double stranded DNA copy of the viral RNA, and this gets integrated into the host DNA. So this forms a part and parcel of the host DNA, and which is now called as a provirus. Now this provirus gets transcribed and gets translated. It gets transcribed into mRNA, and mRNA gets translated into various proteins. Now these proteins are nothing but the structural proteins and the enzymatic proteins which are responsible to form new virions. अभी देखा है हमने कि एक वायरस पार्टिकल के अंदर क्या होता है. There is nothing but a DNA and RNA and is surrounded by a glycoprotein. So it just requires nothing but a nuclear material and a structural material. So all these mRNAs having translation processes into various viral structures, that is protein structures, and the it forms a part of the genome of the nucleus as well. And here the enzyme protease is very active in the final stages of assembly and of virion particles and the budding process into new virions. So there's basically 10 steps you get to see. One is a binding, second is the entry, third is the uncoating, then the enzyme action, then you have the transcription processes taking place, and the DNA copy being formed before that, then the translation process, and then the protease action which is involved in assembly, and then comes assembly and the budding and the release of new virions. So this process also happens with DNA virus, wherein the enzyme RNA polymerase is involved, and you have the DNA getting converted to RNA with the help of RNA polymerase, and the RNA is getting translated, mRNA is getting translated into various proteins. But in case of RNA viruses, you have the RNA virus, the viral RNA itself serves as its own mRNA. So you have the direct process, no enzymes are getting involved. The direct process of viral mRNA getting translated into proteins are happening. So understand the difference between how the three different viruses they interact. Now this image shows of HIV virus. So here you have the enzyme RNA retroviruses also acting upon. So in this table, we get to see some host cell structures that can function as receptors for various viruses. Now, for example, the first host cell structure is T lymphocyte, the helper T lymphocytes on which CD4 glycoproteins are present through which the HIV causing AIDS virus can interact. Another entry point for HIV causing AIDS viruses are the CCR5 receptor for chemokines, MCP1 and RANTase. MCP1 is monocyte chemoattractant protein 1 and RANTase is regulated upon activation, normal T cell expressed and secreted. So these are uh, the cells where the receptor for CCR5 are present, and again the HIV AIDS virus can interact. Next is the CXCR4 chemokine receptor for cytokine SDF, that is a stromal cell derived factor 1. Again, where it serves as an entry point for the HIV AIDS virus. Rabies virus interacts with the acetylcholine receptor located on the skeletal muscle. Glandular fever virus interacts with the C3D receptor on the situated on the B lymphocytes. The T cell leukemia viruses they interact with the interleukin 2 receptor on the T lymphocytes. Infantile diarrhea viruses they interact with the beta adenoceptors, whereas adenovirus, which causes sore throat, conjunctivitis, and the T cell leukemia viruses, they act through the major histocompatibility complex molecule. So coming to the antiviral drugs, we have got antiviral drugs which act by three main mechanisms. The first mechanism is it's an entry point wherein the virus is able to penetrate the host cell. The second point is involving the enzymes, which leads to transcription and translation of proteins. And the third point of action is through boosting the immune mechanisms. So let's see one by one and the drugs involved. One is inhibition of penetration of the host cell. So we are aware that at one place where on the cell membrane of the host cell, the virus employs its own mechanism to fuse with the cell membrane and have its entry into the within the into the cell. So you have drugs like amantadine, rimantadine, which inhibits the uncoating and is very effective against influenza A virus. They also interfere with the fusion of the viral membrane and the endosome membrane, and also in the later stage of assembly and release of new virions at the host cell surface by blocking the ion channels. So look at this, it works well at 2 points. One is the entry point and the other is the exit point where new virions have been produced involving the host cell machinery and they are about to be released. So at both the places these drugs they act. Gamma globulin contains antibodies against the virus envelope and can neutralize some viruses and prevent their attachment also to the host cells. So you have drugs like amantadine, rimantadine and gamma globulin coming under this class, which inhibits penetration of the host cell by the viruses at 2 points. One is the entry point and the other is the exit point. The second classification is by drugs which will inhibit the transcription of the viral genome. Now, the classical drug here is acyclovir. Now, acyclovir is it's the most effective selective antiviral therapy uh, for such treatments, and this agent is basically a guanosine derivative. See, this acyclovir is getting converted initially to monophosphate and then to the triphosphate form involving the enzyme called thymidine kinase. Now, this drug is 30 times more potent against herpes virus enzyme than the host cell enzyme. That's why the host cells are not much affected. Acyclovir triphosphate is a guanosine derivative, and being a guanosine derivative, it gets converted to its triphosphate form, and this uh, plays foul with the enzymes involved in the DNA synthesis. This selectively inhibits the viral DNA polymerase. So viral DNA polymerase, when it is taking one of the nucleotide as a guanosine, and you have a false nucleotide being formed by acyclovir, so it interprets the acyclovir triphosphate as its nucleoside, and it incorporates, leading to false DNA synthesis. So this is effective against herpes virus. The most of the unwanted effects associated with antiviral drugs are very minimal, except for very few cases it is very selective. The local effects like local inflammation and renal dysfunction is common, along with nausea and headache. The other drugs are ganciclovir, and ganciclovir is also a guanosine derivative which is phosphorylated and incorporated into viral DNA, suppressing its replication. This is used in cytomegalovirus. The other drugs in this class are valacyclovir, which is a prodrug of acyclovir, and famciclovir, which is metabolized to penciclovir, the active compound in vivo, and has action similar to acyclovir. So you have got prodrugs also, which are prodrugs of acyclovir, and you have drugs which gets metabolized to other drugs which are having similar actions to that of acyclovir. So other drugs coming under DNA polymerase inhibitors include vidarabine. Vidarabine is an adenosine derivative and is a relatively selective inhibitor for viral DNA polymerase. It is effective against herpes simplex and varicella zoster virus. Next drug is tribavarine. It is similar to guanosine derivative. It is a guanosine derivative and is thought to interfere with the synthesis of viral mRNA, and therefore it can inhibit many DNA as well as RNA viruses. The other drug is foscarnet, which has a different mechanism of action. It inhibits the viral DNA polymerase by attaching to the pyrophosphate binding site, and it is effective in cytomegalovirus infection. Foscarnet is a drug which can cause serious nephrotoxicity, and therefore it is used as a second line drug in the treatment of cytomegalovirus. Cytomegalovirus can be treated very effectively by ganciclovir like drugs. The another mode of action under inhibition of transcription of viral genome is the reverse transcriptase inhibitors. Now, since this enzyme is found in um, uh, uh, this, it is involved in the drugs which are used in the treatment of AIDS. Um, you have a very popular drug like zidovudine and other non-nucleoside reverse transcriptase inhibitors or protease inhibitors coming under this class. So reverse transcriptase inhibitors, the first drug under nucleoside reverse transcriptase inhibitors is a drug called zidovudine, which is an analog of thymidine, and it is very effective in HIV and AIDS treatment. What does this do is this drug gets phosphorylated by cellular enzymes to its triphosphate form, in which it competes with equivalent cellular triphosphates, which are essential substrates basically for the formation of proviral DNA by the enzyme viral reverse transcriptase, that is viral RNA dependent DNA polymerase. Reverse transcriptase enzyme is also called as RNA dependent DNA polymerase. So this enzyme, what does it do? Mammalian alpha DNA polymerase is relatively resistant to the effect, and gamma DNA polymerase is sensitive and this may be the basis for the unwanted effects like anemia and neutropenia observed with zero OD. Resistance to antiviral action is observed because of rapid mutation of this virus. The other reverse transcriptase inhibitors are zalcitabine and diadenosine. They are used in combination with protease inhibitors. Another subclass of this category is a non-nucleoside reverse transcriptase inhibitors. They are chemically diverse group of compounds and they bind to the enzyme directly near the catalytic site and denatures it. So it is directly very toxic to the enzyme. Now, which drugs act under this class? Lamivudine, stabudine, nevirapine, dilaviridine, and loviride. So far we have seen about enzymes, but here the drug is directly acting on the enzymes, reverse transcriptase enzyme, and they do not come under the category of nucleoside RTIs. Well, the third subclass of category of drugs coming under inhibition of transcription of viral genome is the inhibition of post-translational events. So these are the events which are taking place after the translation of mRNA into proteins have occurred and involving the enzyme protease. So this enzyme is a very important enzyme which helps in conversion of the freshly formed viral proteins into functional and structural proteins. So these drugs are collectively called as, which act on this enzyme, they are collectively called as protease inhibitors. So protease inhibitors, they inhibit the cleavage of nascent viral protein into functional and structural proteins. So which class of drugs are they? Which examples of drugs are saquinavir, ritonavir, indinavir, and neflinavir. So these drugs are used in combination of reverse transcriptase inhibitors that will have a synergistic action. And in the treatment of HIV, the treatment of AIDS, the mode of treatment is regimen is you have to have at least minimal two nucleoside reverse transcriptase inhibitors with either a non-nucleoside reverse transcriptase inhibitor, which we have seen in the earlier slide, plus in addition to it, or one or two protease inhibitors. So nucleoside RTIs is must, two drugs. In addition to it, either a non-nucleoside RTI or one or two protease inhibitors should be included in the treatment regimen of HIV. Now, there are other mode of action also of drugs which are acting on a slightly in a different way, like other drugs such as HIV integrase inhibitors. Now, integrase is this reverse transcriptase enzyme involves this another enzyme called as integrase, which integrates the part of viral DNA with the host DNA, and this part of action is inhibited by HIV integrase inhibitor, and the drug is ratilgravir, and the chemokine receptor antagonist. They can also act at the receptor level. Chemokine receptor antagonist called baroviroc, and the drug, the HIV fusion inhibitor, and the virus is not able to enter the cell through this drug, enfuvirtide. So these combination of drugs with the other classes is also preferred in this treatment mode in HIV. Okay, so the last class is immunomodulators. Immunomodulators are basically drugs which will enhance the host defense. We are aware of that. They're called as interferons. Interferons are a family of inducible proteins. Now, inducible proteins means what? They will lead to the production of certain proteins which will have the specific action. So they are a family of inducible proteins which are synthesized by mammalian cells, and they are generally produced commercially using recombinant DNA technology. Now there are at least three types of interferons: alpha, beta, and gamma, constituting a family of hormones involved in cell growth and regulation. Interferon gamma, which is also termed as immune interferon, is produced mainly by the T lymphocytes as part of the immunological response to both viral and non-viral antigens. Interferon alpha and interferon beta are produced by B and T lymphocytes. macrophages, and fibroblasts in response to the presence of viruses and cytokines. Interferons bind to specific ganglioside receptors on host cell membranes. They induce in the host cell ribosomes the production of enzymes that inhibit the translation of viral mRNA into viral proteins, thus halting the viral replication process. So, what do they do? Basically, they will lead to production of certain enzymes that will inhibit the translation of viral mRNA into viral proteins. This is the main action, and thus they can stop the viral replication process. Okay, so we have seen about three mode of action of antiviral drugs. One which will involve the at the fusion process that will inhibit the drugs which will inhibit the penetration of the host cell. Second is the inhibition of transcription of viral genome, in which we have seen about DNA polymerase inhibitors, drugs like acyclovir, ganciclovir, or vidarabine or tribavirin type of drugs. Then the reverse transcriptase inhibitors like zidovudine or stabudine type of drugs, and inhibition of post-translational events like protease inhibitors. And the third class, the immunomodulators. So majorly three classes of drugs as antiviral, but with specific mode of action. These drugs, they don't have a very high risk of any uh, uh, adverse effects, except for some which are really troublesome, like we have seen about foscarnet causing nephrotoxicity, or paresthesia like effects with protease inhibitors, or apart from that, very common gastrointestinal disorders like nausea, vomiting, diarrhea is commonly seen with most of the antiviral drugs. Okay, so I hope you all have understood this topic, and this topic is pretty important from your exam point of view also.